Since 2009, the Imagine Solutions Conference has gathered annually in sunny Naples, Florida, to take a fearless look at the world's biggest problems and offer big solutions to those problems. Driven by a profound respect for learning, the conference is founded, funded, and managed by active, involved community leaders. From economics to education, military to medicine, arts and culture to gender and race and other subjects of the utmost importance in our way of life and in the future of our society, the conference brings together top leaders from all over the world to address transformative ideas and connect intellectually curious people who want to make a difference. A global perspective, but a national focus, an unflinching look at the biggest problems facing our world today and how exactly we can try to solve them. Regular attendees of ISC have become accustomed to thought-provoking presentations by thought-provoking people. And in these unprecedented times, we are happy to bring you that intellectual vigor and spirit once again in 2021. Welcome back, everyone, to day 2 of 6 days of incredible learning. Imagine Solutions 2021, virtually. To paraphrase the famous WINS news radio slogan, give us 90 minutes and we give you the world. That's the goal here today. And today, something that to our knowledge has never been done before. For the first time ever, we bring together 4 science superstars from the world of biomedical research. But what makes them special, beyond their remarkable accomplishments, is that they all work within 1 mile of each other in Kendall Square, Cambridge, outside Boston. We call it the Biomedical Genius Cluster. And this is one cluster where you need much more than a mask to feel at home. Some history now. Back in the early 1970s, Kendall Square at the back of the MIT campus in Cambridge was vacant land, parking lots, warehouses. And today it is home to 9 of the most important biomedical research labs in the world. Within 2 miles are 200 biomedical companies, and among more than 1,000 in the Boston area. So what have these companies done? Well, start with thousands of patents, technology licenses, tech transfers. Those breakthroughs have powered hundreds of biomedical startups, including Moderna, the COVID-19 vaccine pioneer. The scientists, the researchers, and entrepreneurs at these companies and labs sit on the boards of almost every public biomedical company in the country. And today, we are honored to have 4 science superstars from the Genius Cluster, though they're all too humble for words like those. First is Robert Langer, perhaps our country's most honored scientist. He is a co-founder of Moderna, one of just 11 institute professors at MIT. He has 34 honorary degrees, 1,300 patents, and is the founder or co-founder of 41 companies. Next is Dr. Penny Heaton, CEO of the Bill and Melinda Gates Medical Research Institute. Dr. Heaton and her team are changing the world by developing products to fight malaria, tuberculosis, diarrheal diseases, among the globe's biggest killers. Dr. Sangeeta Bhatia is the John J. and Dorothy Wilson Professor at MIT's Institute for Medical Engineering and Science and Electrical Engineering and Computer Science. Dr. Bhatia holds patents for both clinical and biotechnological applications of engineering principles. And if you understand all that, you're a better person than I am. In her spare time, she is a tireless advocate for gender equity and a co-founder of the Boston Biotech Working Group. And finally, bioengineer James Collins. He's the Termeer Professor of Medical Engineering and Science and a Professor of Biological Engineering at MIT. He runs the Collins Labs at MIT and Harvard. He's affiliated with the Broad and Weiss Institutes and was the first bioengineer to win a MacArthur Foundation Genius Award. Our distinguished scientists will be led in conversation by Bob Buderi, veteran journalist, author, editor, and entrepreneur.
We have a wonderful group. It's beyond world class, I was thinking. It's universe class group of people here today, innovators, entrepreneurs, pioneers, and we're really excited to have you here. The audience has your bios, so I thought that reading about what you are is not the same as you describing in your own words who you are and what you do and what really ignites you. So I thought we'd start off with just a little bit, a minute or so from each of you about how you would describe yourself. So I'll just say, Penny, why don't you kick things off?
Sure. Well, thank you. Thank you, Bob. And it's great to be here with everyone today. Yeah, I would say I'm an infectious disease nerd, and I've been an infectious disease nerd since I was a kid. And that eventually led me into a career in pediatric infectious diseases. And after my fellowship, I went to the Centers for Disease Control in Atlanta, and I learned how to work up outbreaks like the pandemic we're going through right now. But I had a study in Kenya where I saw firsthand kind of what happens when poverty collides with infectious diseases. And that changed my life forever. So I've been looking for solutions ever since. And that's why I am at the Bill and Melinda Gates Medical Research Institute in Kendall Square today, still looking for those solutions.
Fantastic. Bob?
Yeah, so I'm a chemical engineer by training, but when I finished my degree, I did something that chemical engineers didn't do at the time. This was the 1970s, and chemical engineers generally went to the oil industry, and I ended up going to Children's Hospital and was the only engineer there. But that got me really excited about what you might be able to do, in terms of engineering and chemistry to medicine. And so I've spent really my whole life doing that. And actually, I've had the great pleasure of interacting with Penny through the Gates Foundation, Sangeeta and Jim, and you, all in different ways. So it's a pleasure to be on this panel with all of you.
Great. Well, we're definitely going to dive into what everyone's doing. Sangeeta, you're next on my screen.
Great. Well, hi, everybody. Let's see, I'm the daughter of immigrants, and I was raised outside of Boston, went to public schools, and then trained to become both an engineer and a physician, and have really been drawn to that kind of intersection my whole career. My specialization in engineering is nanotechnology, which really came about because we were all trying to make our computers faster and make computation portable. And those teeny tiny circuits turn out to be really useful for transforming medicine. So that's kind of the intersection that I live at. I work at MIT now and I run a lab. So I get to be an inventor and a professor and a 5-time entrepreneur. And also there are not as many women in engineering and entrepreneurship as one would like. And so I've become a diversity advocate as well.
Yeah, we're going to talk about that as well. And Jim.
So I'm Jim Collins. I'm a bioengineer at MIT, and I come from a family of engineers and mathematicians. My dad is an electrical engineer who was part of the aviation industry, and there's a lot of talk about moonshots in today's scientific world. He was part of the moonshot, and his team developed the altimeter for the lunar module for the Apollo 11 mission that landed the first men on the moon. And I was brought up in this environment where we saw all these cool technologies my dad was bringing home, and became inspired to go into what became bioengineering when I saw both of my grandfathers become disabled. One lost his vision and the other had a series of strokes. And I saw all these great things my dad's companies and colleagues were creating to shoot stuff up in the sky and shoot stuff out of the sky. And I saw absolutely nothing being done for these two men who I love very much to restore function and allow them to live their lives fully, and became intrigued about this idea of using engineering in biology and medicine, really before bioengineering got established, but initially worked on devices for my first decade to restore function, including to stroke patients. And at the end of the last century, which is funny to describe it that way, but the 90s became intrigued to what engineers could do in molecular biology and helped launch what became synthetic biology, which is a big focus for our lab now around creating living diagnostics and living therapeutics. And we also have a big mission to advance engineering approaches for the discovery, development, design of new antibiotics. And we recently launched what we're calling the Antibiotics AI project, of using AI to really address one of the existential crises facing humankind, which is the emergence of resistance against a backdrop of a drying up pipeline of antibiotics, to see if we can use the power of AI to address those gaps.
Wow, well, that's, we're going to get into these fascinating fields a little bit as we go along. I think some of the themes that already jumped out is that at least with Bob and Jim, you're both engineers. And I guess, Sangeeta, do you consider yourself an engineer as well?
Yes. Yeah.
Yeah, and these are fields that not so long ago you didn't think of in medicine. And yet you've applied them to medicine and to healing, to diagnosing, and in some cases, Jim, like to creating new organisms, right?
Yeah, it's interesting. I think that the introduction of engineering into medicine really finds its roots on the heels of World War II, as technology was being advanced for the military, but also you had soldiers coming back that were missing limbs, had lost function. And so you saw engineering applied to medicine still from the device side, the inorganic, where you saw prosthetics emerge. You then saw a move towards using electrical engineering to get after cardiac space. And so it was still largely more on the inorganic element up until, say just a couple decades ago, as Bob and others really begin to advance tissue engineering. So really becoming the wet side of engineering on advancing us now to create new tissues that could be advanced. We got intrigued of now moving, could you go even lower? Could you go subcellular and begin playing with living cells and their components? And I think what we're going to see in this century is the emergence of engineering biology as a dominant technology, meaning of using really biology as a substrate. Biology is not yet an engineering discipline and still gets in the ways of our best thoughts and best designs, but I think we are increasingly moving toward organic and interestingly the interface of organic and inorganic to address some of the biggest challenges we face as humans.
Bob, why don't you pick it up from there and talk about the tissue engineering and then how you see this this intersection playing out?
Yeah, well, I thought Jim gave a great description. I think that well, tissue engineering, just to go over that, I mean, the idea we had was that maybe someday you could create, and I did this with one of my collaborators, Jay Vacanti, who was a surgeon, back in the early 80s, is that maybe what you could do is make new tissues and organs from scratch, really any tissue or organ, so that, and the idea was that maybe you could have a template like a polymer template and have different cell types on it. And, that would ultimately lead to some forms of what are called artificial skin, also to organs and tissues on a chip. Now it's gotten to be a pretty broad field, like Jim said. And, Sangeeta, we work on it. Sangeeta, I mean, lots of people work on it. And, so and I think it's, a lot of companies have started on it by all of us. And so that's it's been exciting.
Sangeeta, pick it up from your point of view, because I know you're into a lot of this as well.
Sure. Yeah, I think, building on Jim and Bob's comments, one way that I like to think about it is in length scale. So if you think about the progression of engineering over time, what Jim was describing in prostheses and devices, those are large objects, instruments like MRI machines. That was sort of the origins of engineering in the field. And as the engineering technology became able to fabricate smaller and smaller objects, it turns out that length scale, so a cell is 10 microns and a receptor on a cell is 10 nanometers. And as the field allowed us to be able to make objects that are 10 microns and 1 micron and 100 nanometers and 10 nanometers, all of a sudden we had these tools that could talk to biology in its own language. And we could build with these tools. So you see now 3D printers that you can build with living cells as objects. We can make nanomaterials that can package RNA and enter into the body. And so it's really been the progression of engineering to these teeny tiny length scales that now has allowed us to be kind of on the same playing field with where the difference between health and disease lie in the body.
Penny, when you're at the Gates Medical Research Institute, do you get involved in the in these intersections of engineering and medicine and so forth, or are you at a different kind of vantage point?
Yeah, so at the Gates Medical Research Institute, we're looking at developing vaccines, drugs, monoclonal antibodies for diseases that affect the poorest, so TB, malaria, et cetera. So we are really just waiting with bated breath, if you will, for all of the advances that Bob and Jim and Sangeeta are making. Because the diseases that we're working on, let's take tuberculosis. We don't understand tuberculosis from an immunologic perspective. We know that 90% of the people that get infected, they never go on to develop disease. Only 10% go on to develop disease. But by the way, since a quarter of the world's population is infected, that means 1.5 million deaths every year. But we don't understand what's happening immunologically. So what we're looking at and what we're following very closely is the progress that's being made, like the organ on the chip, the lymph node on the chip. Can we begin to use those tools to understand, immunologically what's happening with these diseases, and then capitalize on that data to make better solutions? I mean, the TB vaccine that we have in studies now, it's having its 100th birthday this year. That's crazy, right? But we're only in the last decade or so have we been able to actually have the tools to understand the mechanism of action and how it works. And so we're learning that, oh, well, if we give this vaccine as a booster in tweens and adolescents who got this vaccine originally at birth, we can boost the response and potentially prevent them from being infected. It's like we are taking advantage of the new technologies to figure out what we should have known a long time ago and then take it to the next level.
Penny, we see something similar in cancer research,
Yes.
where the tools are teaching us about the disease at the same time that they're offering new solutions. So for example, in cancer, we now have immunotherapy, which we didn't have just a decade ago, where you turn the immune system on cancer. And that can be both understood better with some of these tools, including things like AI that Jim was mentioning, as well as manipulated better, for example, with nanomaterials that can stimulate the immune system. So I think you're right that we learn as we go, and the tools kind of fall on both sides.
Yeah, and I think a lot of those advances, Sangeeta, that you're talking about with cancer vaccines, or the combination of, immunomodulators, checkpoint inhibitors, and cancer vaccines, I think, what we're doing from the infectious disease side is really watching that space really closely. For TB, could it be that ultimately the answer will be an immunomodulator, a checkpoint inhibitor, with or without a vaccine? So we want you to hurry up so we can hurry up and actually have more impact more quickly.
Now, it might be worth just defining a little bit more in lay terms what you mean by a checkpoint inhibitor and these other terms.
Sure, I can take a stab at that. I think so. Your immune system can fight infections with something called antibodies, which B cells make, and something called killer T cells, and they kill infected cells. And those same killer T cells actually can surveil your body for cancer cells. And what happens is the cancer figures out how to hide from those T cells. And so there are these new drugs that we were talking about, figure out basically how to unveil the tumor so that the T cells can find it and kill it. And so, what Penny was alluding to is, can you also then use the same idea to find a hidden infection, like a TB organism that's hiding inside the lung? That's
great. Well, listen, and we're going to go back, and hopefully, we'll come dive into some of these things you're working on. This is the Biomedical Genius Cluster Panel. So I wanted to ask a question for people. People know about, I think, worldwide, they've heard about Silicon Valley, of course, but they know less about the Boston Kendall Square cluster. So I wanted to, try to have a little fun and ask you guys about what it is that makes this area so special. But first, maybe we could just do a little ranking, 1, 2, or 3, where the Boston Kendall Square Medical Research Cluster ranks in the US in your view. Can you hold up 1 finger, 2 fingers, 3 fingers? I was pretty sure that was the answer I was going to get. What about in the world? Okay. So now let's talk a little bit about why that's true. You know, Bob, you've been here the longest. You've been here for, I guess, since the 70s, right? Or
Yeah, 1970. Yeah.
yeah. So what is it in your view that makes this area so special, and why do you justify that #1 ranking?
Well, yeah, this is going to sound probably very arrogant, but I mean, if you look at rankings, you know, I think every year MIT is ranked as the #1 college in the world in engineering, and Harvard's ranked as the #1 medical school in the world, you know, most of the time. So, I mean, you have, you know, and in addition to those two, there's lots of other fantastic places like BU and Brandeis and Tufts and Northeastern, and, you know, I could go on and on. Wellesley, I mean, there's just so many great places. So you get all these incredibly bright people, and they, you know, spin off some things. They like being in Boston and stay here. And I think what's happened is because of having the universities and the medical schools, everything else comes with it. You have the venture capitalists come here. You have the law, you know, big law firms helping. I mean, so it goes. At least that's what I think. I mean, it's, you know, those two places, you know, help make Cambridge and Boston unique.
Kind of feeds back in on itself. Others dive in there.
Yeah, I think, Bob, I think your phrase, both Bob's, I think it's a positive feedback system. So I think the young talent and seasoned talent from the universities, coupled with the venture groups and the strong law firms, feeding local companies that then attracted larger companies. So most major pharmas have their research efforts or a research effort in Cambridge and some in Boston, but universities, med schools have just built this community. And I'll speak that I think Cambridge and Kendall Square in particular is really quite unique. And I speak from experience in that I spent my first 24 plus years at Boston University in Kenmore Square. And a little over 6 years ago, moved 8,000 feet into Kendall Square over to MIT, into a dramatically different world where I literally am walking out in the street and you're bumping into venture capitalists or key entrepreneurs. And there's just so much more activity going on, and that the density and close interaction of the community is not been reproduced in other spaces. And I think it's very much to be said for the geographical cluster we now have in Kendall Square. And I think back to my undergrad days in the 1980s, Kendall Square was barren. I never felt comfortable walking around. Luckily, I was a competitive runner, so I had no worries. I could outrun almost any person coming after me, but it was a barren set of abandoned lots for the most part that have now, it's hard to find open space that you could put up a new building.
I agree actually completely with Jim that there's something about the density which is really magic. You know, that gives you that serendipitous bumping into people, meeting them in the elevator, seeing them at the coffee shop. I moved further away, Jim, after I trained. I went all the way to California. And I realized that I was really addicted to the density of Kendall Square and ideas. And it is actually an intellectual destination for people around the world, right? I mean, I think we pull talent from everywhere to this like tiny little space, and then they bump into each other and feed off each other and create that feed forward loop that Jim's talking about.
Yeah, I want to, Penny, you just founded, or you opened the Medical Research Institute just a few years ago. You had your pick of places around the country, at least, if not the world. What brought you here?
Yeah, and it's exactly what everyone's been saying. So the Gates Medical Research Institute, we launched just a little over 3 years ago. And we did look at several places in the US, including, you know, Bay Area, Philadelphia, the DC area, et cetera, and ultimately selected this area in Kendall Square for all of the reasons that you all have been saying. I mean, we at the Gates Foundation believe that all lives have equal value and everyone deserves the opportunity to live a healthy and productive life. So when thinking about that in the context of developing products against diseases that disproportionately affect the poor, the idea is you want to bring the latest cutting edge technology and the best talent to these diseases. And so where else better to do that than, you know, the biotech capital literally of the world. And I often give the example, my very first day going into the WeWork space with my one administrative assistant and a bunch of job descriptions when we were getting ready to open it. I was on the T and I was sitting across on the red line, sitting across from someone, and I was listening and I was like, that's a statistician. Oh, they're designing a study. Oh, it's an adaptive design. Oh, that's really cool. Now, I never heard the product and I don't know, you know, exactly what the study was about. But this is exactly the kind of place where you can hear that conversation while sitting, you know, on the subway. So yeah, that's why we chose to be here, and the partnerships that we've developed just in the short time that we've been here, it's been amazing. And we are really excited about the progress that's being made.
I was, you know, it's said it's on the Kendall Square Association website, the most innovative square mile on the planet. I actually found the guy who coined that term, and he likes to. He's for Boston. He worked for Boston Consulting Group in 2009 or so, and he said it was only because he didn't have a chance to survey the rest of the universe that he phrased it that way. But I mapped it out, and it's actually a square half mile, Kendall Square, even under the expanded definition. It's so dense that you really do run into people. I think one of the other things that's kind of alluded to is the starting of the companies and the ability. Now, Bob, I think you're up to 41. It was like 2 new companies while I've been talking to you.
My students are working hard.
But you said to me that virtually all of them at least start out in Kendall Square. You can walk to them, you can consult with them. Maybe talk more about that ability to kind of go back and forth between the world of academia and the world of entrepreneurship.
Yeah, well, I think, first of all, I think an awful lot of the students who work with all of us, Jim, Sangeeta, and myself at MIT, they work hard on their projects, and a lot of them want to see them make their way to people to do the kinds of things that Penny said. And so, almost every company I've been involved in starting, I've done with the students because that's something that they want to do. And the great thing about the Cambridge area and being, all the things being close, like Lab Central and other things, is that you can walk to them back and forth. And so it's like Jim and Sangeeta said, there's this giant infrastructure that just keeps getting bigger and better. And so I just think it's been a wonderful career for them. And so that, and they've really enjoyed doing that. They want to make a difference in the world, and it's been a great way for them to do it. And, for me, it's been really a pleasure to work with them and to work with, the investors and the business people to try to make things happen. So, we did it, and, and we'll probably keep doing it, as long as I'm able to.
Yeah, maybe Sangeeta, 2 of your new companies started in Lab Central, I think. Maybe you could just take people like out your door of your Koch Institute lab, how far you have to go to interact with the company you founded.
Yeah, sure. Yeah. So one of the companies is called Glimpse Bio, and just to riff off of what Bob was saying. So, the students, had the aha moment, and we published a paper, and we thought that if we wanted to take it to patients, like Penny was saying, that we needed to do a startup. And, so you could just be in the building and walk around the building. Like, I could go up to Bob and say, like, is this a good idea? And if it is, who should I call? And then you get an introduction to an investor, and then you find the people, the business people to help you. And so that all can happen very quickly without going very far. And then once the company was established, we put it in Lab Central, which is at 700 Main Street. We're at 500 Main Street. And then they licensed the patents from the technology licensing office, which is just another sort of quarter block away. So, you know, it's all incredibly co-localized.
That ability, and Jim, I don't know what it was at BU, because you started companies when you were at BU as well. But a lot of universities still have that kind of ivory tower feel to them. And maybe MIT had a head start by not having that. Its whole makeup was about applying engineering to society. But is there something about that attitude that it's okay to go from academe to industry that's different in this area?
You know, I think we've seen a large scale change in attitude across the field within the academic space. I started as a professor in 1990. And at that time, it was still viewed as kind of dirty if you were doing translational work, and that the pure were doing the basic work. That changed through the 90s, in part from pioneering work by Bob, in part by people seeing more and more on opportunities. And I think there's been a very healthy attitude change in as follows, that now in even academia, the emphasis is increasingly on impact. And certainly we can still have impact through discoveries and publications and citations and through training our students, but increasingly, even within the academy, there's the question, how many lives have you touched? And how many lives have you touched through maybe introducing a product or a platform? And people are viewing that as a very positive outcome and not simply profit motive. And I think MIT, Harvard, I think Kendall Square has played a huge role, obviously, not only from the early biotech revolution, but now this next wave that's come on its heels. And I think we're increasingly seeing professors also being accepting that their young trainees don't need to be simply clones of themselves to become other professors, but that there are viable, innovative, very rewarding routes through other nonprofit and for-profit entities, which is great. But I'll spin it the other way as well. I think in some fields we're seeing a problem, and as follows. You know, I really respect all that Bob's done and view him as a true role model, and that he stayed in academia, and we have the flexibility where he can spin out a number of companies, be involved, but not necessarily run them. And we're seeing in other fields like computer science, AI, deep learning, that the field is making it offer so attractive to basically diminish who's still in academia. So they're eating the seed corn. And so we're not training this next year. We don't have enough people to teach those. There are new models coming out where people now are splitting appointments with Facebook, Google in a particular university. But I'm worried that some fields are pulling away the folks that can train the next generation.
Just by dint of the how much money they can make, or?
I think it's two things, Bob. So we've always had the battle with industry of what talent we can recruit, whether at the postdoc level or as professors. We never compete on salary, but we compete to give them freedom and to do innovative, creative things. But now these large companies are offering these amazing resources and the freedom to do creative, innovative things. So now if you're going to be paid 10 times more than you would as a professor, and you can still run a group of 20 people doing cutting edge blue sky work, and you can publish, you'd be an idiot not to take that option. And that's the model we're running up against. And fortunately, I think some of the bigger groups are now recognizing that they need to give us back talent, at least part-time, to continue to train the next generation.
Well, I think what Jim said is right, you know, and I actually don't, it's kind of just thinking of a funny story, you know, MIT wanted me to go, I think it's called Renaissance Capital. I never really, you know, so I was giving this, like, and I really didn't know what it was. And but you want to always help MIT and stuff like that. So I went there, and I really should have read up on it, because I really didn't know what it was. And I remember getting there, and they had this like fantastic lunch, 40 kinds of sushi and everything. And I spent like an afternoon talking to different people. And I thought, my God, these people were all former professors at places like Berkeley and Caltech and all these, and they were incredibly smart. They asked all these questions, and then they were really like incredibly good questions. And so I tried to ask them a question or two about what they were doing, but they sort of didn't want to explain it, which I felt, okay, you know, I'm there to help MIT raise money. You know, so then, you know, I remember after we had the dinner, I did ask one of the women, I said, well, why did you leave Berkeley? Because, you know, it's such a great school, and, you know, to come to this place in Long Island. And she sort of said, well, I guess it's about the money, you know, and so I remember, like, when I was flying back, I decided to check out, you know, or be in the car. I was looking at my, you know, looked at the internet to look up what it did. And I saw that, like, this was like this hedge fund thing, and they had like an 80% return every year or something like that. So then all of a sudden I understood. And I think that's a whole other area, you know, people, you know, who are just incredibly smart going into hedge funds, and they do incredibly well. In fact, I was, you know, on the board of trustees of Cornell. I've been on other sort of charity boards, and, you know, there's a very large percentage of hedge fund people at all of them because they make so much money. And I think that, in fact, I remember one of my daughter's friends, it was at Yale, and she said something like that 25% of the undergraduates, their goal was to be a hedge fund manager. So I think what Jim said is absolutely right. I was only expanding it to other areas as well.
There's a new book out, Bob, called The Man Who Solved the Market by Greg Zuckerman, that tells the story of Jim Simons, who was the founder of Renaissance Capital. And well, Jim, you know, he was a math professor at Stony Brook, who used machine learning to make predictive models of the market to create this incredibly successful hedge fund.
Yeah, no, it's funny because Greg, I've gotten quite a few calls from him. He's now writing a book on COVID vaccines and Moderna.
But even math models aren't very good. They just had 20 to 30% loss this past year. So they're not.
Is that right? Right. Wow. So yeah, so I went two or 3 years ago. That's good.
Well, you know, I think maybe a counterpoint, you know, which is living together with this. I agree, this huge influence on the students, especially in computer science. I have a joint appointment in computer science. You see the students get recruited even out of undergraduate, you know, with these gigantic salaries that Jim and Bob are talking about. I think the encouraging thing that sits side by side with that, which is that this generation of students that's growing up with social media and worldwide awareness are actually are really activists in many ways, and they are really aware of the state of our planet and global health. And so I think I'm really encouraged by, you know, a set of students who are so oriented to impact that they're willing to work on things like global oncology, so cancer care around the world, which, you know, a lot of people don't think about. And so I think it was interesting. I mean, you sort of have to hold these two opposite ideas in your head, but I actually think that our student body has both of them.
Well, Penny, you're in the nonprofit world. What do you see in you know, in terms of the motivations of the young people and so forth?
Yeah, I mean, I think I agree with Sangeeta. I just love what I'm seeing today as far as the generation that's finishing their training and coming out. I mean, if you think about our mission at the Gates Foundation and the Gates Medical Research Institute, you know, to develop products for TB, malaria, enteric diseases, maternal, newborn, and child health. Those problems aren't going to change. We just have to figure out how to identify the solutions, but the problems are there. It's not like we can say, oh, we don't want to work in infectious diseases anymore. We don't want to work on TB. We're just going to set that aside. You know, that remains the leading infectious diseases killer in the world. And so we have to figure out what the solutions are, regardless of where those solutions might come from. So it really does mean working, you know, across the aisle with, you know, we have academic partners, we have, you know, big pharma partners, we have biotech partners, we have public health partners. And that's just an important part of what we do. So I think, and I think that's part of what I see, and what Sangeeta was referring to is kind of the activist side of these, you know, kind of segments of young people coming out. They see the problem and they're going after the problem and thinking creatively about how to break down the silos to find the solutions.
Well, this seems like a good point to ask some questions about the skill set and the talent set of the future, like what you guys see. You talk about your fields that in many cases are the convergence of different technologies. Jim's talked about AI as well, and Sangeeta about computer science. What do you see as the kind of driving forces for the future in terms of what types of skills, what technologies will be coming together, and what students, might need to have to succeed?
I think this is maybe an unexpected answer, that in addition to technical depth, which you absolutely need to be able to sort of synergize with someone in another field, you need to have a place of depth of your own, that more and more we're seeing that our trainees really need to be strong communicators, with each other, with venture if they're raising money, to be leaders, and really important in this moment also with the public, because we need science literacy, we need trust in science. And so, we're really finding that we have to, in this generation of trainees, we have to be deliberate about our training, and have students invest in that conversation with the public, because for so long it was just like publish a paper and talk to each other. And that doesn't allow people into sort of like the beauty and the magic of what we're doing. And so I think, the moment communication is, I think, one of the most important next gen skills.
Very interesting. Well,
I'll maybe jump on that, Bob. To build on Sangeeta's point. I think a few points that are lost on the current generation of young talent that would make a difference is, one, I think content matters. And I think young people need to be willing to put in the time to really have a strong fundamental base understanding across a good number of disciplines, and then make the commitment to become an expert, truly a content expert in the area or areas they want to get into. And related to my next comment is, the young people need to use their best resource, which is their brain, and not the machines we have at the end of our hands, whether smartphones or PCs. And the ability to look up stuff is so different than actually knowing something. And if we're to remain innovative, the best device to be innovative is our brains. And it's to make connections between those stores of content that you worked very hard to establish in your head, to see connections between things maybe somebody didn't see before. Two is, I think communication is absolutely critical. And probably the most important thing most of us could speak to that we got from our undergrad and training, and probably played such a big role in our careers. And related to that, I think that young people need to disconnect from social media in order to improve communication and to be comfortable, to Sangeeta's point, with themselves, and to not always be comparing to what else is happening, and to be comfortable in themselves. And that's then pointing my third point, is that I think young people should put in a habit of spending time with themselves every day, just thinking about interesting thoughts, leaving it open, and being comfortable in that you could spend that time. Maybe you don't come up with anything, but it's taking that time to ruminate inside your head, whether on a walk or a run, listening to music, and doing nothing. That we're always checking everything nowadays on all these platforms. And it's become a distraction. And I'm worried that we're going to have a reduced innovation output from this generation because of the technology that's been created.
Very interesting. You reminded me when I was a little kid and I complained I was bored, my mother told me, you should never be bored, because when you're alone, you're in your own best company.
Yeah,
I thought that was really great. And it helped me to not be bored and to think and those kind of things.
yeah.
Well, I'm going to switch gears a little here. I want to hear, I know the audience will want to hear some of what you're all working on. Some of the 2 or 3 projects that you're working on now that light your fire, or that you're envisioning right over the horizon. So, maybe Penny, you could start things off.
Sure, absolutely. So as I alluded to earlier, our number one priority at the Gates Medical Research Institute is tuberculosis, because of the massive mortality from it every year. 1.5 million deaths. That's like having a COVID pandemic, but every single year. So it's insane. So we're working on both vaccines to prevent disease, and then we're working on drugs to actually treat the disease until a vaccine comes along. So it's such a tough problem. It's not one that you can tackle alone. So on the drug side, on the therapeutic side, we are working with a consortium of 4 pharmaceutical biotech companies and the Gates Foundation, our parent organization, to look at different drugs that can have activity against TB. So for those of you who don't know about it, the way you treat TB, it takes 4 drugs for 6 months or longer in order to treat it. It's just a really hard disease to treat. If you think about an organism that has learned how to live quite happily with humankind, for 15,000 years, it's a pretty clever organism, so it's pretty hard to treat. And so instead of having 4 drugs for months and months and months on end, and they have horrible side effects, what we are looking for is a shorter, simpler, safer drug regimen. And to prevent resistance from occurring, we need to use drugs with different mechanisms of action. So the likelihood of 2 or 3, 4 drugs coming from a single entity is really, really low. So this is one of those big, hairy problems that just requires incredible collaboration. And that's why we're working with this consortium, this pan-TB consortium, to identify new drugs and bring them through the pipeline and to the patients that need them. So we're hoping to actually start with clinical studies of our first novel TB drug regimen by the end of the year. I'm really excited about that. And then, as I alluded to earlier, we also have the 100-year-old TB vaccine. But in addition to that, we have a TB vaccine that we in-licensed from GlaxoSmithKline that is fascinating because it doesn't just prevent infection, but it actually prevents in people who are already infected. It prevents progression of tuberculosis to pulmonary disease and death. And so it's interfering somewhere in that progression. And so we're looking at on the immunologic side, trying to figure out how it works. We already know from a clinical study that was done before that we licensed it that it does work. And so we are getting ready to take that into clinical trials as soon as we get new clinical trial materials made. So those are just a couple of things. I could talk for hours about all the things that we're working on, but really, really exciting stuff. One thing I would like to mention that we're super excited about is the RNA vaccines, that they're using for COVID. Well, before the pandemic, we had already started to look at collaborations on RNA vaccines for tuberculosis and for malaria. And now with the pandemic, maybe one of the silver linings is that those programs can be just greatly accelerated because of all the knowledge that has been found out from the work on the pandemic.
Well, let me just ask, so in all these years, there hasn't been an effective TB vaccine?
There's this.
Why can we do COVID in 1 year? And
great question. That's what I keep asking my team. If we could do COVID in 1 year, we've got to speed up the TB programs. It's fascinating. I mean, I think, quite frankly, because TB was controlled through drugs, different therapeutic regimens in the 1950s, 1960s in the US and Europe in high income countries. Then the funding and the investment in further advances in TB, they just weren't there. And in fact, still the funding envelope globally, when you look at TB research, is just abysmal. And because of that, the advances just weren't made that needed to be made. And of course, when the foundation was formed in 2000, they targeted HIV, TB, and malaria. But the other thing is TB is tough. It's immunologically really complex. So I would say it's really only in the last decade that we've been able to have the tools to even understand the disease to the level that we need to in order to be able to develop vaccines. COVID is so much easier by comparison. TB is really tough.
Well, it seems like a great segue. Bob, you one of the founders of Moderna that made this vaccine that's making that's now getting out. Maybe talk a bit about what the vision was on that, and then some of your current projects as well.
Well, I think Moderna's goal has always been to try to make our messenger RNA therapeutics, and quite a few of the examples of that are vaccines. I think there's 9 or 10. In fact, a lot of the Gates Foundation has been very, very supportive of Moderna, and that's been terrific. And the Gates Foundation has also been very supportive of our lab. So that so when you get to projects that we're doing, that's how I first met Penny, and we have quite a few projects with them. Some aimed at some of the things she's talking about. We're working, for example, on a way of single step. You hear about how you have to do multiple boosters or multiple shots. We're working on a way, again, a lot of this is for the developing world, but it could be useful for our world too, is that you could give we published a paper in Science, for example, that was fully funded by the Gates Foundation, and where we came up with a way, Anna Jakulinik in our lab led this, and where you could give one injection, but that single injection, because of the way we designed the micro or nanoparticles, would have about 12 bursts. And we can time the bursts whenever you want, 1 month, 2 months, a year, and so forth. Another project that we did with the Gates Foundation that actually led to Lindra, which is a company we started. And it's actually relates to something Sangeeta mentioned. I'll have to ask her if she'll know another company like this. The chairperson of the board, the executive chair of the board, the CEO, and the COO are all female. And it didn't happen because the science all came out of our lab that the Gates Foundation funded, but basically they were interested for treating malaria and other diseases. Could we come up with a pill? Because pills you're going to be more likely to take than other things like shots, that could last for a week or a month. So we're coming up with a once a month pill for birth control, a once every 2 week pill for malaria, and so forth. And we are working along the lines that Penny mentioned, ways of Gio Traverso, who worked with me and now is a professor at MIT, developing pills that or systems you could swallow that might be able to have even 100 grams. This is for tuberculosis that she was talking about, not for vaccines, but for drugs. And we're working on new ways of giving micronutrients to the developing world. Aside from the Gates Foundation projects, we have a lot of others. We're also working on pills you could swallow that could deliver proteins and maybe someday even RNA or DNA. And we're working on all kinds of regenerative medicine things, everything from trying to help people who are paralyzed with spinal cord repair, to new ways of restoring hearing, to new treatments for diabetes.
Wow. That's one of the things here is there's this, it's not just an incremental, a lot of the projects you're talking about big changes that really make a huge impact. And I think that's part of the culture in this Kendall Square Boston ecosystem. Sangeeta, why don't you talk about some of your projects?
Sure. Yeah, I think alluding to Jim's earlier point, which is it's helpful to have depth. I've spent my whole career obsessed and in love with one thing, which is the liver. And
not because of alcohol in any way.
Yeah, actually, if you go to a liver conference, the joke is that we're all keeping ourselves in business, at the happy hour. But yeah, so it's a fascinating organ. It's the size of a football. It sits under your rib cage, and it has over 500 functions, and it affects 500 million people worldwide a year. So it's liver disease is what people call a stealth epidemic. You don't feel it always when you're sick, and it's really kind of quietly amongst us. Most recently, there's a disease called fatty liver disease, or NASH, non-alcoholic steatohepatitis, which is going to be the biggest cause of liver transplant. So, it's a sort of underappreciated organ. It's a factory, and we really don't know much about it. And so a lot of the technologies that I've been developing in my lab have been things that allow us to see how the liver is doing, little nanosensors that can enter into the liver and tell you what the microenvironment is like. And then you can you can have a urine test. So it's like a shot and a urine test, and it tells you how your liver is doing instead of getting a biopsy. And that became the foundational technology for a company called Glimpse that was in Lab Central that we were talking about earlier. And then we've been trying to come up with a way to actually replace transplant completely, which, in spite of being an amazing procedure that's life-saving, is actually still really hard to do. It's hard on the patients, and you have to wait for an organ to be available. And so we're making little off-the-shelf implants that you would put in the belly in a minimally invasive procedure that would provide enough liver cells for patients to live without getting a transplant at a kind of a satellite location. And that became the technology for Satellite Bio, which is another Lab Central startup, and is really inspired by Bob and Jay's work so many years ago, where they sort of invented this idea of using cells as living medicines. So I'll stop there, but you know, if you spend some time with me, just like Penny could talk about TB, I could talk all about the liver.
Awesome. And Jim, please tell us some of your exciting projects.
So I'll mention three. So the first is in this idea of wearable synthetic biology. So a few years ago, we showed that you could take cell-free extracts along with synthetic biology constructs, spot them on paper, freeze dry them, and then reactivate the freeze-dried spot sometime later with water or a patient sample, and reactivate the transcription translation processes to create what we then used as paper-based diagnostics, for antibiotic resistance and for Ebola, Zika, and we've done some for SARS-CoV-2. About 2 years ago, we showed that this could be extended beyond paper to clothing of various forms and move towards creating wearable diagnostics for first responders, for healthcare personnel, for military personnel, athletes, detecting pathogens, metabolites produced by the body, as well as if you're exposed to a nasty chemical agent. But in the face of the pandemic, we pivoted the project to develop a face mask diagnostic for SARS-CoV-2. And we are, as I speak, finishing the studies to show that you can have an insert added to a face mask based on this freeze-dried cell-free synthetic biology that can, with high specificity and sensitivity, detect SARS-CoV-2 relative to other circulating coronaviruses, as well as detect influenza, so seasonal flu. So we can give a readout similar to a pregnancy test after wearing the face mask for only an hour or so. And it's a very, very low cost insert that can be added to any face mask. The second project is really on recognizing that much of synthetic biology has been in the bacterial space, but is really emerging strongly in the mammalian space. And I think in the next decade or two will dramatically impact gene and cell therapy. And in the spirit of Moderna and other efforts, we've been focusing on how you can use synthetic biology to help advance the regulatory aspects of mRNA therapeutics, those delivered for gene therapy, but also those delivered in, say, a Moderna fashion, say, with engineered liposomes. And amongst other things, we've now developed mRNA-based switches where you can repress the translation of an mRNA and have it turned on by one or more other trigger RNAs that are present in the cell. And so you can now have cell-specific, tissue-specific activation of your therapeutic RNA, mRNA, that's either, again, delivered, say, with some of the technology Bob has on lipid nanoparticles, or via a viral vector. The third is related to a product I mentioned at the outset, which is around the Antibiotics AI project. So going back about a year and a half ago, Regina Barzilay, one of our colleagues at MIT and an AI expert, we teamed up to create platforms that allowed us to train a deep neural net, initially off of just training data from E. coli, to arrive at a model that could be used as a screening tool against repurposing libraries or large in silico libraries to begin to identify new antibiotics. And we've identified some powerful new antibiotics that are very effective against a broad range of pathogens, including TB, Acinetobacter baumannii, and C. diff. Where I'm excited is we're taking it in multiple directions, but two to note. One is we're now training models against specific pathogens like Acinetobacter baumannii and TB. And second is we're now advancing the models not simply as screening tools, but as design tools. So not simply taking what was learned to then screen it against the library to see, can we find molecules with the properties, but to see what was learned by the model that makes for good antibiotics and use those insights to design de novo novel antibiotics to go against some of the nasty pathogens that are out there.
That's really fascinating. So you think you can definitely see the day when you can design the antibiotic. You can actually just program what you need.
To an extent, yes. You in the end still have issues of toxicity you want to include in the model. You have issues of synthesizability. So we're not in this case doing antimicrobial peptides, which are much easier to program because you can encode them in a DNA sequence. Here we're getting after structural properties. And so it becomes the challenge of ambition versus actuality, in that some of the more interesting molecules may have 100 steps for synthesis, which would make it very, very difficult, if not impossible, to synthesize. But we're hopeful that by taking advantage of the power of what AI affords with now deep networks, deep neural nets, that we can look at a bigger space and chemical space to see where we might be able to go for new antibiotics.
I want to, well, especially in the time that we have left, I want to ask Sangeeta about. We talked about the talent of the future, but you spoke briefly, and Bob mentioned also the company with all women executives. Sangeeta, you've been very active in something I've been helping with a little bit too, the Future Founders Initiative, and maybe you could talk about that and what it says to the future of innovation.
Sure, yeah, I'm happy to. So this is an initiative that I started together with Susan Hockfield and Nancy Hopkins. And Bob, you've been helping along with Harvey Lodish and Bob Langer and many others. And the conversation really that we started was around the observation that women academics were not starting companies as much as their male colleagues. And so we first set out to quantify that. It turned out that under 10% of the 250 plus companies in 7 departments at MIT were started by women, even though they've been 20+% of the faculty for some amount of time. And then in some departments, for example, the disparity was really dramatic. 56 companies came out of one department, 54 by men, 2 by women. So, you know, what that means for the innovation ecosystem, we calculated was that we would have 40 more companies if women were starting companies as often as their male colleagues. So we sort of banded together and said we have to do something about this. And the Future Founder Initiative is basically one of those things, and it has a bunch of prongs. One of them is a boot camp that Harvey Lodish and I ran in the fall virtually to try and inspire women. So 400 people across Boston have signed up for that boot camp. And these are basically stories of people who have started companies, the kind of the inside scoop of how you do it, and also what it does to your science and how it can increase impact. We've been working with venture capitalists to engage women in the ecosystem, because really access to capital is a really important bottleneck in starting a company. Today, 2.7% of VC dollars go to women-founded companies. And several VC firms in town, including Polaris and F Prime, have signed up to do a pledge to increase the number of women on their board of directors to try and get them engaged in the system. That doesn't make you a founder, but if you're on the board or in the scientific advisory board, at least you're in the room, you know what that looks like. We've gotten the deans involved at MIT and Harvard to do like a sabbatical program to release women from teaching so that they could go be in residence at some of these VC firms, and many, many more things. And actually through this conference, I met Cecilia Conrad at the MacArthur Foundation, and she and I are talking about how to do this in a scalable way so that we can sort of create a future founder action toolkit that we could replicate across the Boston ecosystem and then also across the country. There's very similar data that's now come out of Stanford.
When you talked about the 40 missing companies, that was over what time period?
Yeah, it's tricky. So it's probably over 30 years. The way we did it is by looking at who's on the faculty now, right, and looking backwards. So it doesn't account for people who were here and passed away, like Sue Lindquist, or have left for other reasons. So it's just a rough estimate.
Right. And one of the things that's really, I think, important is that there is this feedback loop, and it's not necessarily that women are deliberately excluded all the time, but you're in this system where you have to know, you have to be comfortable with the field, you have to have access to the people, and you're partly trying to break down those kind of barriers in that way.
Yeah, that's right. Just to be an accelerator. You know, what we find in conversation with our women colleagues is sometimes they don't know that they have a company. They don't know that they have an invention. And then even if they have the inkling that they do, they don't know where to start. And so just connecting them to people who have done it before, like there's so many wonderful mentors in our system, is at least part of the process.
Well, as we wrap up here, is there anything, again, you hinted at it in some of your answers or your comments, but what is maybe some of the good things that might be coming out of the COVID pandemic for medicine more broadly? Penny, I know that's something you talked about a little bit. Maybe you can just give a quick hit on that, a minute each.
Yeah, I mean, as I already said, I'm super excited about RNA vaccines for other infectious diseases. You know, we now have proved the potency of the platforms. I think that, and I think the way we do clinical trials, we're doing a COVID trial right now that's almost entirely enrolled remotely using telemedicine. It puts the patient right at the heart of the clinical trial. So I think that's another bit of progress that could be made with COVID.
Sangeeta, maybe a quick one from you because you may have to jump off.
Sure. Yeah, you know, it's funny. I guess everybody looks at the vaccines with a different lens. What I see, in addition to the RNA, is the nanoparticle package. And so as a nanomedicine person, like I see this year as a huge victory lap for all of the sort of fundamental investments we made in nanotechnology, that these particles were really ready to rise to the challenge and now have been de-risked in millions of people so that we can repurpose them for other things.
Fantastic. Bob?
Well, I agree with both, you know, both Penny and Sangeeta. And I think what Sangeeta said, I just want to emphasize, so many people, you know, really put down nanotechnology when some of the things didn't work as well. But, you know, this clearly just shows if it wasn't, you know, the RNA vaccines wouldn't work if it wasn't for the nanoparticles, they'd be destroyed immediately. But I also, in addition to that, I can just tell you what I hope. I think there's no question that what Penny said is right. I think messenger RNA, and of course, being a founder of Moderna and being on the board, I feel like that's great. I think that's a really important thing for the future. But what I hope, and I don't know that we're there yet, you know, I think some of the things that Jim mentioned about face masks, and I would say more broadly about medical devices, I just hope that really the world realizes just how important all these different things are, you know, everything from molecular biology to medical devices and diagnostics. And, you know, the fact that so many people in the US alone died because we really didn't have companies in place to do masks. We didn't have companies to do in place to do diagnostics. And the only reason really why, I'm not trying to brag about Moderna, but it's totally because investors and other people put, you know, it's not what people have said. It's not because, I mean, it's great that the government put in lots of money, you know, this year. But if if the investors didn't put in money for Moderna to build the manufacturing plants many years ago, you know, we wouldn't have those vaccines either. So I think all of this is really important and shining a light on what should be done.
Jim?
I'll give two things. One, picking up on Bob's last point, I'm really encouraged by the interest now in diagnostics from investors, from clinicians, from companies. And I think there's a major gap that's been identified. I don't think that gap's been appropriately filled yet. I hope to see it better filled in the coming few months, but I think points to just the challenges we have and the needs in that space. And two, at a higher level, I really think the pandemic will serve to excite young people about considering challenges in infectious diseases, showing that these problems are important, they have impact, and they're tractable. When I look to see the young hotshots that enroll at MIT that want to do life science work, prior to the pandemic, there were two dominant areas, neuroscience and cancer. Two areas of great import, two areas with tremendous exciting problems. But I think we'll now see three on the heels of the pandemic with infectious diseases coming up. And I very much applaud and hope that applaud Penny's work and hope that TB will help slot into that, given that this is a pandemic that is costing us 1.5 million lives every year and has been for decades. And we need more young talent going after TB and related problems.
I did want to get in one more question about the kind of schism that we have, where there has been such distrust of medicine and science. Is there any way you see to overcome this, to bring more people to accept the, anybody have any thoughts?
I'll chime in, Bob, as a small point to start. So I've been concerned on the mistrust of science. And, you know, it's interesting. I think a couple points. There's the famous trite phrase of that everyone's entitled to their own opinion, but you're not entitled to your own facts. And I think we need to get back to that to really establish what's fact and what's opinion. But we also, in that regard, also, I think, have to better educate the public about what science is really about. So I think science as a term is being misused. That science is not the opinion of a scientist. It's actually a rigorous study that was conducted and then analyzed. And I think a lot of people don't appreciate that we disagree all the time. And that's at the heart of it, that we don't disagree on the facts, we disagree on the interpretation and implementation. And I think we as well have fallen into a trap where we kind of rally and you create a cult of personality around a few number of folks, when in fact it should be a community that's arguing and talking about it. And I think we have fed some of the mistrust a bit, and we need to acknowledge when we've been wrong. We need to acknowledge when things have changed. And I think in doing so, we can build trust for when the data are strong to support a recommendation. And I think we've helped, unfortunately, to create some of this outside of the lunacy that's occurring in social media and folks purporting to put out, you know, false news. But we as scientists, I think, need to be very honest about how do we function as a community.
Yeah, I mean, to add to that, I think it goes back to some of the things we were discussing earlier. In order to really address this issue of, you know, people not trusting science and us not having an agreed on set of facts, I think we need to look at the intersection of disciplines again between the expert psychologists and social media and how people learn today, how they actually form their set of facts. And we need to bring them together with the scientists in order to come up with solutions. So I think, you know, in the vaccine field with vaccine hesitancy, it's such a huge problem. And we brought together a group where we had experts from, you know, all of the social media platforms, Facebook, YouTube, Google, et cetera, with the vaccinologists. And it was amazing, some of the ideas that came about, but those aren't groups that normally come together. So somehow we have to force that union in order to find these solutions.
That's fascinating. I'll follow up with that. Bob, last thought from you on this?
Yeah, no, I just think, and you're in a good position to go over what I'm going to just say, but I think the media could do a lot. I mean, all kinds of ways of doing a lot. You know, in some cases, I think they've done a really bad job of scaring people and misinformation. And I mean, places that are, you know, would probably be considered by a lot of people reputable. And I think on the other side, and I think there's so much that the media could do to, you know, popularize science and really, you know, help. And so I think there's a lot of opportunities in the media to do a lot of good things and not a lot of bad ones.
Yeah. Hey, well, listen, you guys, this has been fantastic. Thanks. I'm sorry to keep you over a little bit here. Thank you for your time. I look forward to when we can get together in person sometime again.
Yes, thank you.
Thank you so much.
Now let's take a look back to the history of Imagine Solutions for another in our series of aha moments.
You know, you either wake up in the morning and you're interested in whatever is going on. I love these two little words, and I don't think we pay enough attention to them: over and next. When something is over, it is over. On to next. And if there was a hammock in the middle of those two words, that would be the best definition I know of living in the moment.
What a terrific conversation that was. And that, folks, concludes day 2 of Imagine Solutions 2021. We'll see you right back here tomorrow, same time, same place. I'm Tyler Matheson. The 2021 virtual Imagine Solutions Conference is made possible through the generous support of our sponsors.